Hey, today we're going to talk about electrical physiology. So we're going to cover excitable cells. We're going to talk about the electrochemical gradient that we need to make action potentials, then resting membrane potential and action potentials. So excitable cells are cells that are able to produce and respond to electrical signals. This obviously includes neurons, muscles, right? So your skeletal, heart, and smooth muscle, and then secretory cells like your pituitary or your islets of Langerhans that release insulin. And so those are important as well, right? Why electricity? Well, think about it. If I wanted to move my big toe and I had to rely on diffusion, um, how long would that take to get the impulse from my brain to my big toe? Okay, so a long time. Why do we use electricity or why do we use separation of charge? Because it occurs much faster. So electrical impulses take about nanoseconds to milliseconds to get where they need to go. In order to do this, though, you need separation of charge. You need an electrochemical gradient. So we're going to separate ions in different amounts across the plasma membrane, and that's going to give us potential energy. So the ability to do work, right? The electrochemical gradient is the sum of all electrical and chemical forces acting across the cell membrane. This gives us our potential energy. This is due to concentrations of charged particles. If you take a look at the picture inside the cell, you have negative A, which stands for negative proteins. You have potassium, which is a positive 1 inside the cell. You have chloride, which is a negative 1 outside the cell, and then sodium, which is a positive 1 outside the cell. Take a look at these ions. So sodium, and I would like you guys to draw a picture. So draw inside the cell and outside the cell. Actually, how about just draw a cell, draw a circle, and then inside the cell, note that sodium is 15 millimolar, right? And outside it's 150 millimolar. Potassium is 150 intracellular, and extracellularly it's 5. Chloride ion is 108 extracellularly, and negative proteins are 65 intracellularly. Okay, so, and I'm drawing my picture right now, so you should be drawing your picture as well. So sodium outside is 150, and then inside is 15. And then potassium is 150 inside and 5 outside. If you did the dry lab, the Mastering AMP, the Neurophysiology dry lab, you'll notice that these are a little bit different concentrations. So for the dry lab, they have potassium as 15 extracellularly. Obviously, every textbook is going to have different concentrations of ions inside and outside the cell. The one that's pretty constant, though, is potassium extracellularly. That actually doesn't change much. So this is what I would like you to go with a reference for potassium. Okay, so the electrical gradient. How do we get this electrical gradient? Just so that you're aware of this, an electrical gradient develops when there are more positive or negative charges on one side of a membrane than on the other. These ions are going to move towards the area of opposite charge. So positive ions would move towards the negative side, and negative ions would move towards a positive side. A chemical gradient develops when there are more ions of the substance in one area than in another. So for example, there's more sodium outside, and so sodium wants to move down its concentration gradient inside the cell. Take a look at your picture while I'm explaining this, right? So sodium is higher outside, it wants to move inside the cell. So sodium has a driving force to want to move inside the cell because of its concentration gradient and because it's a positively charged ion. It wants to go into a negative area. Okay, so this is where you really need to pay attention. The electrochemical gradient is established. Let me change my little clicker to my pen color. Let me see what pen color I have it as. Let's do it as something that you can see. We'll do it as black, I guess. So the electrochemical gradient is established So what does that mean? So the electrical chemical gradient is established by your sodium potassium pump. So your sodium potassium pump is what sets up that separation of charge. How does that pump work? You're going to pump out 3 sodium and you're going to drag in 2 potassium per 1 ATP, right? So take a look at this pump. This pump, if you look at the picture, let me change my pen color. Let's do something that's going to stick out. We'll do red. So if you take a look, this pump, this sodium potassium pump, is going to pump out how many sodium, right? So 3 sodium. And how many potassium is it going to drag in? So it's going to drag in 2 potassium per ATP used. So if we're using ATP, is this active or passive? Okay, so if we're using ATP, excuse me, this is active transport. So because we're using ATP, we know that this is active transport. Sorry, I'm trying to get this to work. Right? And because we're using ATP, it's active transport. And we're kicking out 3 sodium and we're dragging in 2 potassium. So if this is active transport, if we're using ATP, and we're kicking sodium out, where is sodium highest? So let me rephrase it again. If we're using ATP to kick sodium out, where is sodium the highest? Good, so sodium is the highest out, whoops, that's funny, out here, right? Now look at the picture that you drew with the concentrations, the 150 for sodium on the outside and 15 on the inside. Does that align with what you know? Yes. Okay, and then if we're dragging potassium in, where is potassium the highest? Good, potassium is the highest here. So potassium is highest here. How do we know potassium is highest here? Because we're using ATP to drive this pump. And so it has to be active transport. And if it's active transport, we're pumping sodium where it's highest and we're pumping potassium where it's highest, right? Otherwise we wouldn't need ATP. Here's a little animation that you can use to look on it or to understand it better. Let me actually go over this with you. So I'm going to draw a picture. Let's see, pen. I am going to pause the recording for a second and I'm going to come back to it. Let's see, pause share. Oops,